Hi everyone, welcome back to Biology 101. This is Amrita and today we have two topics, that is multiple allelism and pleotropism. Last class we learned about codominance, right, where the progenies represent the characters of both the parents. Now this is a different topic, multiple allelism. That is, usually in humans, no, we are diploid, right? So it can we can see only two alleles for a particular character, but now in this case there are more than two, that is 3 alleles maybe or 4 governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. When we are making a population, like in a city, we are calculating about the alleles and all, no, that time only we can see more than 2 alleles. Usually it's diploid. So that is where this multiple allelism comes. Now again, multiple allelism, we have very good example of ABO blood grouping. Even in codominance we saw, right, this is the same example. Now ABO, why? Because it is controlled by a gene I, and this gene I has 3 different variants, that is IA, IB and small I. That is why multiple allelism. What is basically multiple allelism? That is more than 2 alleles govern the same character. Okay, so that is multiple allelism. Now next, pleotropism. Now what is pleotropism? A single gene may produce more than one effect. This is the opposite of multiple allelism, because in multiple allelism many alleles govern the same character, right? Many alleles govern the same character, but here a single gene, one single gene may produce more than one effect. Okay, so one single gene may produce more than one effect. Example we can take is a synthesis of a pea. Let's say there are two alleles. For example, starch synthesis in pea seeds. Okay, it's controlled by 2 alleles, that is capital B and small b. Now let's say if B, that is capital B and capital B, are coming together, they are homozygous, right? Homozygous dominant, which means we have large starch grains produced if both capital B and capital B come together. If small b and small b come together, they are homo, but they are smaller, that is they are recessive, right? So they come in less efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B, we can also, there is another example for this, like pleotropism, no, a single gene may produce one or more effect. Now we have seen this both come under size. Now if we say, if capital B and capital B, after maturing, what happens? After maturing, the BB seeds are round, that is capital B and capital B become round, small b and small b become wrinkled. Now why we call it as pleotropism? Because of greater than one effect. One is the size, now this is about the shape. So shape and size controlled by same B, capital B and this small b. This is a gene, right? So only one gene controls more than one effect, that is, one is size and then second is shape here. Now also one more thing in this example we can see is incomplete dominance, right? Incomplete dominance, why? What is incomplete dominance? The progenies will be showing the in-between character. So if there is BB, if there is a capital B and small b combination, then what will happen? The starch produced will be in the intermediate size, right? Intermediate size. So if starch grain size is considered as a phenotype, then from this angle we can say alleles show the incomplete dominance because of capital B and small b. So therefore dominance is not a autonomous feature of a gene. Like autonomous feature means it is not what is the meaning of autonomous is independent, but here dominance is not autonomous means it's not independent, which means it is dependent. It depends on the gene product and the production of a particular phenotype. It depends on how the product comes out. It is not solely dependent, it's not solely on a feature of a gene, right? So we have pleiotropism and multiple allelism. Thank you.